Welcome everyone to this WaterTalk. It's my pleasure to introduce to you Professor Karen Kidd this afternoon, who's going to deliver the WaterTalk. Dr. Kidd holds the Jaroslawski Chair in Environment and Health in the Department of Biology and School of Geography and Earth Sciences at McMaster University. And we're very honored to have Professor Kidd here with us today. She's a very well-known academic figurehead, both inside and outside of Canada. And I have to admit that even before I had heard of the Water Institute at the University of Waterloo, back in my days in Europe, I had already heard of Professor Kidd's groundbreaking whole lakes experiments at the experimental lake area in Canada to improve the understanding of the risks of exposure to hormone mimics to fish and their habitats, and the timing and severity of impacts of estrogen on aquatic organisms. And this is a key characteristic of the research conducted by Professor Kidd's lab, whole systems experiments and studies to understand how pesticides, nutrients, and other stressors affect freshwater food webs. Before joining McMaster in 2017 as the Jaroslawski Chair in Environment and Health, Professor Kidd lived in New Brunswick and worked in the biology department and the Canadian Rivers Institute at the University of New Brunswick St. John, where she was a professor of biology and held a Canada Research Chair in chemical contamination of food webs. Before that, Karen spent 6 years with Fisheries and Oceans Canada in Winnipeg, Manitoba, as a research scientist. In 2017, the international pharmaceutical industry honored Professor Kidd's groundbreaking work with an International Environmental Award, and she's furthermore on various advisory and editorial boards, too many actually to mention, but most importantly perhaps the editor of the journal Environmental Toxicology and Chemistry, and she was a guest editor 13 times in the proceedings of the National Academy of Science, PNAS. And the title of Professor Kidd's WaterTalk today relates to her amazing record of groundbreaking research, What Happens When the Fish Go on the Pill. Please join me in welcoming Professor Kidd to the floor.
Thank you very much for the generous introduction, and it's a real pleasure to be here, so thank you to the Water Institute for hosting me this afternoon. I'm going to be talking about a study that started when I was a research scientist with Fisheries and Oceans Canada. It was my first real job, and this was my first big undertaking as a young scientist. So it's been going on for a while now. Before I get into the story, I'll tell you, I just wanted to acknowledge, you'll hear me say we a lot, and there's a lot of people I had the pleasure of working with on this experiment. So a number of them are with Fisheries and Oceans Canada, the big list at the top there, but we also had collaborators with the International Institute of Sustainable Development, with the universities of Lakehead and Saskatchewan and Manitoba and Kansas, Environment and Climate Change Canada, as well as the US EPA. So there's a huge team of people involved in this research. So the story I wanted to share with you today is a story about pharmaceuticals and municipal wastewaters, especially estrogens. And I'll start by telling you why we did a whole lake experiment, what were we thinking about in the late 1990s. And then there's 3 chapters. So the first one is about the main character in the story, the fathead minnow, what happened to that species once we started adding low amounts of the synthetic estrogen in the pill to the lake. Then the second chapter is about what happened to all the other species that live in the lake once we saw impacts on the fathead minnow. The third chapter is about what happened when we stopped putting the pill into a lake. Was there recovery of the system? And then I'll end with some of my thoughts about what our results mean for other wild fish living downstream of municipal wastewaters. So the story starts in your toilet, it starts in my toilet, and that's because when we're consuming pharmaceuticals, we excrete them in our urine and feces. When we flush the toilet, those pharmaceuticals are traveling through our wastewater system, hopefully to a municipal wastewater treatment plant. But a lot of the pharmaceuticals and the natural hormones that we produce are not completely broken down during that treatment process. And so in the 90s, there were studies like this one coming out of the UK showing widespread disruption of male sexual development in these rivers. And in some parts of the UK, reaches of rivers, there were 100% of the males that were feminized. And what I mean by that is that these males had smaller testes. They were producing egg yolk protein precursors called vitellogenin that you normally only see in females when they're developing their eggs. And they also were developing this condition called intersex. So they were producing eggs in their testes. And obviously this generated a lot of concern, a lot of follow-up studies about what it is that was the fish were getting exposed to that was causing this feminization of the male fish. And the detective work narrowed it down to mostly the natural estrogens that we excrete, the estrogen that's in the birth control pill, as well as compounds that block the action of testosterones. So that soup of estrogens, anti-androgens is what the male fish were getting exposed to, and that was causing them to become feminized. So this work in the UK motivated a lot of studies worldwide to look for feminized male fish. Do we have them in Canada? Yes, indeed we do. So we see them in the South Saskatchewan River. We see them in Wascana Creek. We see them downstream of Montreal, and we also see them right next door in the Grand River in the work by Mark here at the University of Waterloo. And there's a common thread to all of these studies in that these are species that are small-bodied, so they tend to live in a very local habitat. Their exposure is higher than the large-bodied migratory species. We also know they're from areas where there was poor wastewater treatment, low dilution of the effluent, and a lot of people. And when you look more closely at some of the fish that Mark's group has sampled here, when you open them up, this intersex condition that I've talked about is sometimes very obvious. You don't need a microscope to see the presence of eggs in amongst the white testicular tissues. But this is what it looks like if you stain the tissues and then put them under the microscope. You can see these round objects in here. Those are eggs in amongst the sperm cells. And so in the Grand River, before the wastewater treatment plant was upgraded, there were over half of the males that had this condition of intersex. So why did we decide to do a whole lake experiment? Well, we knew a number of things at the time. We knew that males were becoming feminized in these rivers. We knew that it was being linked to the presence of estrogens in the wastewater effluents. But the big question that we wanted to address, and one that you can only address with a whole lake experiment, is so what does that mean for the sustainability of the fish population? So can those males with eggs in their testes still successfully reproduce? Or is there going to be an impact on the abundance of the fish? So we chose to do this work at the experimental lakes area. And if you haven't heard of this facility before, it's world famous for its ability to manipulate whole lakes. And it's done a lot of high impact research that has had amazing impacts on our legislation around acid rain, reductions of phosphates in detergents. It's a research station that's located in northwestern Ontario, and it's an area that was set aside for research. So there's no development in the watershed, there's no commercial or sports fishing. I also call it the zombie research station, and that's because in 2012 the federal government cut the funding for this facility. And then it was taken over by the International Institute of Sustainable Development, and they're doing an amazing job of running it. So it's a facility that's come back to life under their direction. And this is a map of some of the lakes and showing you some of the experiments that have been done. So there are a lot of diverse types of experiments that have been very impactful in legislation, changing policies, et cetera. And here's some pictures. So this just gives you a sense of the area that it's in. There's no development in the watershed at all. Lots of lakes as part of this facility, and it's a year-round research station. So there's residences and labs, and it can house about 60 to 70 people each year. This is the lake that we chose to do our whole lake experiment on called Lake 260. And the main reason that we chose it is because it has the fathead minnow in it. And that's a species that we knew a lot about from lab studies. So we knew how it would respond to this synthetic estrogen. But we also chose it because it has the lake trout in it, and that's a top predator, a fish-eating species. And Lake 260 is deep enough to have a trout species in it. And I'll just mention, because this will come back later on, is that it's got one small lake upstream and then quite a large lake downstream of the lake itself. And I'll talk a bit about how a number of fish species responded to the EE2 additions, but focus most of the story on the fathead minnow. So this is our experimental design here. We started the work in 1999 and spent the first 2 summers doing baseline work. So what's in the lake, how many are in the lake. We had to do some small-scale experiments to figure out how much of the synthetic estrogen to add. And then in the spring of 2001 until September of that year, and again in 2002 and 2003, we were adding low parts per trillion levels of this EE2. Our target was 5 to 6 nanograms per liter. And as you'll see shortly, right away we started to see responses in the fathead minnow and other fish species. We monitored the lake for those 3 years of estrogen additions, and then for a number of years after that to see if the fish populations recovered. And certainly at the same time, we were also collecting the same kind of information on reference lakes, because we wanted to make sure that the responses that we saw in our experimental lake were because of the EE2 additions and not because of natural variability from climate change, for example. So we had to add this synthetic estrogen 3 times a week because it's broken down by bacteria and sunlight. It's not a persistent compound. And just to give you some idea of the scale of our study, so if you look at the number of women in Canada that are on the pill, that translates to what we're adding was about 10,500 pills worth of this estrogen every day. And then we're dosing 3 times a week for about 5 months. And so that equates to what's used in a city of about 240,000 people, just to give you some perspective on the scale. This is a picture of the addition. So what we would do is measure out the mass of the estrogen that we needed. We would dissolve it in a bit of methanol, and then we would pump that solution into the propeller wash of the boat and drive the boat around the lake. It would dose the lake in about 15 minutes, and then the waves would do the rest and mix the EE2 horizontally and vertically. And then we had to measure it every week to make sure we were achieving our target concentrations, and the overall seasonal averages were about 5 to 6 nanograms per liter. So the first chapter of my story is about the fathead minnow. This is the most exciting one. And we focused on this species because it's known to be very responsive to estrogen exposure. It's a short-lived fish. It reproduces only once after it matures at the age of about 2, and then it dies off. It's a species that's widespread in North America and an important prey species for a lot of larger fish. So what happened to the fathead minnow once we started adding EE2 to the lake? Well, this is a graph of the egg yolk protein levels in males from our experimental lake in blue before we started adding the EE2, and from our reference lakes. And then these bars are what we saw once the EE2 additions began. So right away, within about 6 weeks, that's the first bar, we saw an amazing response at the biochemical level, and that the males were taking up the EE2 through their gills. It was going to their liver cells and stimulating their liver cells to produce this egg yolk protein. So clearly they were responding at the biochemical level. What happened at the tissue level? Well, this is a section from a male fish just before spawning. So it's been stained and sectioned, and we're looking at it under the microscope. And that's what the sperm cells in the males normally look like at that time of year. They're full of spermatocytes. And this is what the males look like the summer after. So basically after a year of EE2 exposure. So there's a lot of fibrotic tissues in their testes. The sperm cells were in an earlier stage of development. And then this is what we saw 3 years later, was this condition of intersex, like we first found in the UK. So these are this is the presence of early stage eggs amongst the sperm cells. So they were responding at the biochemical level, at the tissue level. What happened at the population level when they were not able to develop their sperm cells as normal? I'll start by showing you a size frequency distribution for fathead minnows from our reference lake. So these bars, all the panels are the different years that we were in doing the study. And what these different panels will show you, the white bars are the young fish that hatched that summer, and then the gray bars are the adult population. And what you can see over time is that some years the young fish make up a higher proportion of the population than others. So some years are better reproductively for this species than others. And then if you look at the numbers that are in the panels, those are our catch per unit effort. So that's the number of fish we caught standardized to the net time. And in our reference lake, you can see that some years our catches are higher than others. And that's just another characteristic of this kind of species, that they have good years and bad years depending on food availability, et cetera. So that's what our reference lake looked like. And this is what our experimental lake looked like over time. So first fall of 2001, after our EE2 additions, we had good reproduction. And then the second and third year of additions, we had a reproductive failure. So there was only a handful of very small fish that we caught, very young fish, and then the adults just kept getting bigger as they were surviving and not reproducing. And eventually the adults too died off, and we ended up with a near extinction of the fathead minnow from the system. So going back to our $1 million question, this graph showed us, or answered that question, that the birth control pill in a lake can cause a crash and impact the sustainability of a fish species. And this graph is showing the data in a slightly different way. So this top panel, those are young of the year that we caught in the spring. The bottom panel, those are adults from the spring. And if you just look at the black symbols in both those panels, you'll see a big drop during the years of EE2 additions for the young of the year, and then a drop later for the adults because they survived a bit longer and eventually their numbers died off too. So you'll see this decline of 99% in the population abundance. So why did the fathead minnow decline so dramatically? Well, we don't really know, but it could be a whole bunch of different things. You saw that they weren't developing their sperm cells as well as they normally should be. What also happened is the males lost their external maleness. So if you look at the picture, here, the top fish, that's a male fathead minnow. And the reason they're called fatheads is because during spawning season, they get a fat head. And they also develop dark banding, and then they get bumps on their nose, tubercles. And I guess that makes them sexy for the female fish. But the fish underneath is a female, except when we were adding the EE2 to the system, the males lost their external maleness. So they all look like female fish. And that probably caused some confusion during reproduction. The males guard their nests, and we looked at the number of eggs at the nest and their behaviors. I'll show you that shortly. But they were producing fewer eggs, and the eggs were at an earlier stage of development. And we did some work looking at the behaviors. So this is work that Paul Blanchfield and his team did where they snorkeled the shallow waters of the lake and some reference lakes. They did some underwater video of the males guarding the nest and compared this behavior before and after the EE2 exposure. So they looked at things like aggression, courtship, and nesting. And what they found was quite opposite of what we would have expected from lab experiments. So this is just the nest defense data for Lake 260 in the black circles, and then our reference lake in the gray. And when you expose fathead minnows to EE2 in the lab, they become less aggressive. But surprisingly, what we saw here is that they became more aggressive. So this is a good example of sometimes how you can't always translate lab data to field scenarios. And then I'll just point out here too, they went back in 2011 and they found the behaviors were back to normal. So chapter 2 is about the other species in the lake. So once the fathead minnow population crashed, what happened to the organisms that it fed on or the lake trout that fed on the fathead minnow? It's a shorter chapter, and I'll just cover some of our results quickly, but we went out every week or every couple of weeks or every month, depending, and collected samples of the water column using this Canadian Tire tube special sampler. Canadian Tire has had a lot of my money over the years. It's a great place for field sampling equipment. And so we looked at algae and bacteria and zooplankton in the water column and how they changed over time. And then we also looked at the insects that were emerging from the shallows of the water using these emergence traps. And before I show you some summarized data, I just wanted to show you some of the massive amount of data that this team collected. And these are biomass data for very small plankton that live in the water column, and they're important prey for small larval fishes. Just to show you that we have a lot of biweekly data over a long period of time. And I'll show you the collapsed summer averages shortly. And what I'll show you is how the rotifers responded, how the crustacean zooplankton responded, how these dipterans, the Chaobarus, responded, and those are zooplankton predators, so they feed on crustacean zooplankton. And then finally, how the emergence of those insects, which are mostly chironomids, responded. And what we saw was an increase in the rotifers. We saw an increase in crustacean zooplankton. We saw an increase in Chaobarus. And then we also saw an increase in the littoral insect emergence. And this increase could be either because there's more food for them to eat or because there's fewer predators around. And what we found is that there's no change in the bacteria or algae available to these lower trophic levels. So we think their increase is because the predators that were feeding on them were declining. So it was an indirect response to the collapse in the fathead minnow and the fathead minnow's exposure to EE2. So what happened to the other fish? Some of them we did the same kind of detailed studies for as the fathead minnow, and others we just looked at numbers and changes in populations over time. So the suckers here, they too produced a lot more vitellogenin than the males did, but there was no evidence that their egg or sperm cell development was affected. And when we looked at the population, so Lake 260 is the black symbols, our reference lake is the white symbols, there was no suggestion that the sucker numbers went down. We looked at the pearl dace, and for those, they too produced a lot more vitellogenin, the males and females. They showed delays in egg cell and sperm cell development, and then the males also developed that intersex condition in the first year after EE2 addition started. And what happened to their population? Well, they're shown in the black symbols here. You can see that their numbers went down during the EE2 additions, but we can't say it's because of the EE2 exposure, because our reference lake population also collapsed, which was unfortunate. Slimy sculpin, well, we didn't look at them at the biochemical or tissue level, just at the population level, and in the second and third year of additions, we saw low abundances of this species as well. And then finally, for the lake trout, they too responded at the biochemical level by producing more vitellogenin than normal, but they did not show any impacts on their egg or sperm cell development. So we were a bit surprised when we saw that their numbers were going down, their biomass was going down in the later years of the study. And we think that's because the small-bodied fish that they prey on normally were declining. So the fathead minnow went down, the pearl dace went down, the sculpin went down. So the trout responded to this decline in food supply by also declining in abundance. So for us, this was another example of an indirect effect of the EE2 additions. So was there any recovery? Well, this is some of the same data you've seen already, the high vitellogenin levels during the EE2 additions, and then when we went back several years later, that was back down to normal in our experimental lake. You've seen all of these images before with the development of intersex in the male fathead minnow. And then this is what the males looked like. Their sperm cell development was back to normal 4 years later, once we stopped adding the EE2. And then this is the population data. This is shown in catches per unit effort. So for the population, it went down, down, down over time, and then there must have been some great mood music in the lake in the summer of 2006 because they had amazing reproduction and recovered completely by the following year. So that was fantastic to see and quite unexpected that they would go from such low numbers back to what they were before the experiment started. So it was great news. So this recovered population, I get the question often, or I had the question often about where are those fish from? And earlier I'd mentioned that there's a small lake upstream and a larger lake downstream, so maybe there was immigration from those two systems. So what we did later in the study is we did some microsatellite work to understand the genetics of the recovered population compared to the original population. And I'll just mention there's beaver dams on the upstream, on the inflow and the outflow to this lake. So physically, it would be really difficult for the fish to move into Lake 260. And there are also no fathead minnow upstream. So that wasn't even a potential source. We went back to our archives and looked at tissues from before the EE2 exposures and compared them to the recovered fish with microsatellites. And these were the data that we got. And if you look first at the green, the black symbols, those are the before and after population of Lake 260. And the closer the dots are together, the more genetically related they are. And what these data told us very clearly is that the recovered population is almost genetically identical to the original population. So there was no immigration from any of our potential source lakes here. So what did we learn from this experiment? Well, there's lots of lessons. The first one was that if you put in low parts per trillion levels of a potent estrogen, you can impact the sustainability of a fish population. The second one was that when you impact the fathead minnow, you can have indirect effects on its predator. The third one was that the reduction in these small fishes led to an increase in fish prey, so another indirect response. And then the last one is that these kinds of experiments are invaluable because we never would have learned this had we done or tried to do this kind of work in the lab. So there's bad news and good news from this experiment. The bad news is that the pill is as effective for fathead minnows as it is for humans, and that it can cause indirect trophic cascades in a lake. And that's important to mention because it's something as ecotoxicologists that we don't often consider, as we often focus just on the direct effects of a contaminant on a species and not what the knock-on effects might be for something that eats it or something that it eats. The good news is, though, that you saw that the fathead minnow recovers. So once you remove the estrogen from the system, you give it enough time, the species rebounded, which is great. So the other question that I was often asked is, should women stop taking the birth control pill? And my answer is unequivocally no. That the answer is in better wastewater treatment. So I'll just show you as you move from primary treatment, which is basically a screening of solids, to secondary to advanced treatment, you progressively remove more of the estrogens out of the effluent and you improve the quality of that discharge to the rivers. So that's really where the answer lies, is in upgrading the wastewater treatment plants to reduce the effluent estrogenicity. And I wanted to almost end, it's not quite the end of my talk, but I wanted to show you some of the great work that Mark's team is doing here at the university, because to me this is another good news story. So they've been doing long-term monitoring in the Grand River since 2007, is that right, more or less? And several of the wastewater treatment plants have been undergoing upgrades. And this is an example from the Kitchener wastewater treatment plant that went to a nitrifying system in 2012. And that means that they're converting their ammonia to nitrates. So you can see ammonia levels drop as that new system comes into force. And what that did is it improved the quality of the effluents in other ways as well. So if you look at the pink circles here, that's the estrogenicity of the effluents before and then after the upgrades. So those upgrades, even though they weren't designed to digest or break down pharmaceuticals or estrogens, they had the amazing byproduct of reducing some of those other contaminants. So they saw declines in estrogenicity, but also some declines in other pharmaceuticals that were present in the effluents. And even better, the fish responded. So as the wastewater effluent improved in quality, the presence of intersex downstream declined. So the red bars, those are the fish that were caught after the upgrades happened, downstream of the discharge, and it took only a few years for the incidence of intersex in the male fish to decline quite dramatically. So I talked a lot about EE2, the synthetic estrogen used in the pill, but we know these effluents contain a lot of other pharmaceuticals. So we have painkillers, we have blood lipid regulators, we have chemotherapeutants, we have anti-epileptic drugs, heart pressure medications, et cetera, et cetera, et cetera. So all of the pharmaceuticals that we're taking as a society for our health and well-being can be found in wastewater effluents. And this is an example from the Hamilton wastewater treatment plant. Some work done by Environment and Climate Change Canada is just showing you all of the different types of pharmaceuticals that are present in the effluents after. So the big question is, well, what does that mixture of pharmaceuticals mean for aquatic species living downstream? And that's a big, outstanding question in our field. I also just wanted to point out this big bar that goes off the charts here. That's a pharmaceutical called metformin that is used for type 2 diabetes. And it's one that's particularly of concern because it's very recalcitrant to wastewater treatment, and it's also not well broken down in rivers and streams, so it's quite persistent and present at very high levels in systems downstream of these outfalls. And it too has been shown to impact reproduction in fish. So what happens to all of these drugs when they're released to the water? Well, some of them are broken down rapidly, others are not. And there's been a lot of different studies that have gone out to look for these pharmaceuticals in fish tissues or in the insects that they eat. And what's more recently been found is that you can have a movement of pharmaceuticals from water to terrestrial organisms. So as these aquatic insects emerge as adults, they can bring these drugs with them in their body tissues, and then spiders and birds that feed on these emergent insects can be exposed to pharmaceuticals as well. So it's not just an aquatic issue, there may be impacts on riparian species as well. And we know from lab studies, so this is just a quick summary of some of the potential effects that these pharmaceuticals may be having on aquatic species. So in the lab, lipid regulators affect lipids in fish. Diabetes medications can affect fish reproduction. Antidepressants affect fish behaviors and reproductive timing in clams. Antibiotics can impact the ability of algae and plants to photosynthesize. And then anti-inflammatories can affect growth, et cetera, in fish as well. And that's because some of the targets in humans, some of the ways that these pharmaceuticals are effective and active in our bodies, those receptors are conserved in fish as well. So they can have the same kinds of effects in fish that they have in humans. So just to summarize some of our challenges, and then I'll mention a few opportunities, but the challenges are that these pharmaceuticals are not broken down completely in wastewater treatment, so they are discharged to our surface waters, and because they're designed to be biologically active in humans, and humans and fish are not that different, they can have impacts on fish health. I've mentioned they're persistent, and the other issue is that there's continual discharge. So even for the ones that are broken down rapidly, there's always new compounds being released and continuous exposure. The other downside is that we currently are only measuring a few of the pharmaceuticals that we're using. So there's a big black box in terms of the other thousands that we haven't looked for in the environment. Are they there? Are they having impacts? But on the flip side, to end on a positive note, there's a number of opportunities that I'm quite excited about. The first one is that we have new wastewater systems effluent regulations now, and they're coming into force. And as the wastewater treatment plants upgrade to meet the guidelines, the new guidelines for solids and ammonia and chemical oxygen demand, those upgrades, as you saw with the Kitchener plant, are going to improve the quality of the effluent for estrogens as well as other pharmaceuticals. And the other opportunity is that, and this is happening more in the US and Europe now than in Canada, but I think that wave is coming, and that's that we're now starting to think of wastewater as not as a waste, but as a resource. And it's a resource of energy for electricity production. It's a resource of water, certainly. It's also a resource of phosphorus. So I think as we change our mindset and we start capturing these resources out of wastewater, we'll too remove more of these microcontaminants like pharmaceuticals and improve the quality of what's being discharged to our surface waters. So I'll just end by saying we've had a lot of fantastic support for this experiment over the decade plus of years from a number of different agencies. All of the EE2 was donated by a pharmaceutical company in Germany. And I'll end by saying thank you for your attention. I'll be happy to answer any questions if you have them. Thank you.
So you said that it's the estrogen that might be the cause. I'm curious as to, like, if we're breaking down the estrogen in our water plants, or sorry, our water treatment plants, how do we know it's not a byproduct that's causing this?
Well, because when you look at the estrogenicity, you're capturing all of the compounds, not just the synthetic estrogen, not just the parent compound, but the metabolites as well. And so we know with those assays that the whole estrogenicity actually decreases as you treat the wastewater. So it's, yeah. Well, yeah, I think you can pretty much get rid of all of the estrogenicity in wastewaters if you treat it with advanced treatment. So ozone is very effective at that, activated carbon.
There's a question here.
Thanks a lot for that presentation. It kind of makes me want to go fishing, but not to eat the fish. Two questions. One is, did you? I didn't see, or maybe I missed. How did the predator fish? How were they affected? Do they get feminized as well? That's one. And the second question is, because they're such a short-lived species, and we have the recovery in the 4th year, right? Do you know if do the feminized males actually recover and reproduce, or is it newborn? I mean, like, how do we control for that kind of phenomenon? What accounts for the uptick?
I'm not sure I understood.
Yeah.
your
So
second
what I
question.
mean is that the feminized males live maybe 3-4 years, and the project ran for that same period of time, and in the meantime you remove the estrogen, right? So who is doing the reproducing? Yeah.
It's a few remnant individuals that were less affected by the EE2, that for some reason they just had a great reproductive year, and they were able to generate offspring to recover the population completely. And that's pretty normal, is that within a species you'll have some individuals that are more sensitive and others that are less sensitive to a contaminant. And your first question was about, now you can tell I've got a short-term memory problem. Oh, the lake trout, yes. So the lake trout, yeah. Yeah. So the lake trout, the males were producing vitellogenin, so higher levels of that egg yolk protein, but they didn't show any delays in sperm cell development. And so that's why when they went down in numbers, we thought that it was likely an indirect response to prey declines and not a direct effect. And then you talked about fishing, and I'll just say that these compounds don't accumulate in fish tissues, and so human exposure to the pill through eating fish is not at all a concern.
There's another question here first.
Okay, sure.
Thank you, Dr. Kidd. That was fantastic. I have
Thank
two
you.
questions. So first, did you notice any downstream effects from Lake 260 on the receiving water body? And second, did you look at the feeding behavior or indirect measures of feeding in lake trout to see if there was a prey resource switch during the time when the prey fish species were not abundant?
So we didn't do any gut content work or stable isotope analyses to see if they were shifting in their diets. So that's an unknown. We were required to monitor the water and the fish downstream to make sure that the EE2 was not getting or not adversely affecting any fish downstream, and we had trouble catching fish in the outflow, but it also went through quite a wetlands area, so likely the EE2, like we couldn't detect any in the inflow of the downstream lake. So I think it was just getting broken down as it moved out from Lake 260 into that body.
Mark?
Karen, I had two questions. So the first one, the lake trout, did you see any indication that the condition was changing?
We did, yeah.
So the condition
It
was...
did go down as well, yeah.
So it supports that hypothesis. And the other question is, have you ever looked for the EE2 in the deep sediments in 260?
We did. We brought sediments back and tried to analyze them for EE2, but had a lot of analytical challenges because of matrix interferences. And the other thing that we did is we brought sediments back and spiked them with EE2. And when you just leave them, the natural microbial communities, they break the EE2 down really fast. And we show that by comparing sterilized sediments to unsterilized sediments. So I think even if it got to the deeper layers, and it did, it was sorbing to particles, and then the particles were settling out to the deeper waters, that the bacteria there would just use as a carbon source, yeah. So breakdown was pretty fast. Oh, hi.
Thanks for the talk.
You're welcome.
Inspired by the question over there, when you said that because you showed that the population, when it rebounded in the fathead minnows, were genetically similar to the pre-disturbance population, if the ones that survived to produce that new population, if you hit this lake again with estrogen, would you suspect that the new population is more resistant if the parents, or many generations ago when you did this experiment, if they were able to continue to produce offspring, would the lake have a different response, do you think, if you did this again?
I think so. It would be interesting to do that. But we've thought about that, and even just bringing them into the lab and seeing how they responded to the EE2 exposures, because there is evidence from other contaminants that fish can develop resistance, I guess, to things like PCBs. So it's possible that they can develop resistance to EE2 as well. Yeah. Hi.
Hi, thank you for the talk.
Thank
I
you.
was wondering if you looked at the recovery of the pearl dace and if it staggered recovery with the fathead minnows, just because I'm thinking maybe predation was lower and then they were able to recover, if you looked at that relationship at all?
We, some of our fish monitoring stopped around 2004. The scientist that was doing that work retired. So, and for the pearl dace and the lake trout, we were using either gill nets or large trap nets. And then when he retired, we only focused on the minnow trap and the species that we were catching there. So, I wish we had that information, but at that point, we were just mainly focused on recovery of the fathead minnow. Yes.
Thank you. That was really interesting.
Thank you.
My question is also sort of related to the sludge or down lower, because of course lake and groundwater, like surface water and groundwater interactions, are you not worried then at all because of how fast the estrogen breaks down of it seeping into the groundwater?
Well, there, the shield, there's a lot of rock, and there is certainly shallow groundwater, but we weren't as concerned about it for this system. And the half-life in our lake was about 12 days, so that's why we had to keep adding it continuously, is because it was broken down pretty rapidly. So I think the EE2 and other estrogens, they can certainly contaminate groundwater in areas where you have effluent discharges and then the groundwater surface water interactions. Yes.
So we have one more question here.
Okay.
Thank you very much for the talk.
Thank you.
Did you see any effect on the female fish? Like you had the intersex in the male testes. Was there any effect of estrogenic compounds on the female at any level?
So the female fathead minnows, the first summer after the additions were started, they had delayed egg cell development, but then the years after that, they were back to normal. And so I think that they had a bit of an adjustment period to this external estrogen exposure, and then maybe they were able to metabolize it after that, and the responses were not as dramatic. So yeah, they did respond initially, but not over the long term.
Any more questions? So I think Professor, there is? Okay, one more.
I was wondering, in light of what you said about this doesn't necessarily mean that women should stop taking the pill and that this is more of a wastewater management issue. Who do you think should be talking about this issue and bringing it forward and making decisions based on this information?
Well, I think it's everybody. I think it's a collective issue. It's those of us that are using pharmaceuticals, the companies that are producing them, and it's the wastewater plant operators as well. And I think that collectively, there's been good examples in Europe where collectively people are taking their unused pharmaceuticals back to the pharmacy for proper disposal instead of flushing them down the toilet. Pharmaceutical companies are recognizing that where they're producing drugs, they need to be more aware of what they're discharging in their wastewaters. So they're looking at improvements in wastewater treatment. And then we're upgrading our wastewater treatment plants, not necessarily for pharmaceuticals, but for other water quality reasons, and that's going to have some really good benefits for reducing pharmaceuticals as well.
No more questions? Then I'm going to hand over to Professor Maurice Dussault, professor in the Department of Environment Science.
Great.
Thanks, Karen. It's good to see you.
You too.
Actually, I have a question.
Okay.
But it's a rock mechanics question.
Uh-oh.
These lakes are on hard rock, so
Yes.
it's very silicious, very little calcium,
Yes.
and they have their own systems because of that, slightly acidic, all these kind of things, very sensitive to. Would you expect massively different results, for example, in lakes that are on, like in Saskatchewan, and because they have a lot more.
Yeah. Well, that's going to change the exposure of the fish to the estrogen. So it's a bit of a debate still. Things like organic carbon in the water can reduce the uptake of estrogens to fish, but also things like increased productivity. So if you have more nutrients in the system, more algal growth, that can absorb estrogens and reduce exposure. So certainly water quality is a big influencer of estrogen uptake into fish. But I couldn't tell you specifically how the prairie systems might behave, other than if they're more productive, then exposures should be lower.
You've got a lot more research to do here.
I guess so.
It's a pleasure to be asked to thank Dr. Kidd. I had the pleasure of working with her on a group of a small committee of people called the New Brunswick Energy Institute for 5-6 years. The Energy Institute is now officially terminated, I think, this week, and the little residual money that was left in the account is going for scholarships at the University of New Brunswick. So that was good news.
Yeah.
And we always had good times in bringing our different areas of expertise to this group, which was certainly not partisan. It was trying to advance science. So there was a groundwater studies done. And Karen did a lot of the baseline work in trying to define the current situation in New Brunswick lakes and streams in the area. And that baseline work is pretty fundamental, as you can see from the 2 years of baseline work done here, to the advancement of the understanding of biota and contaminants and materials transport. So she did a great job there. So on behalf of all of us at the University of Waterloo, and particularly me, I'd like to ask you to join me in once again thanking Dr. Kidd for a very fascinating talk. Thank you.
Thank you, Maurice. Thank you.